The overall goal of this procedure is to visualize and quantify respiratory cilia motility and cilia-generated flow using freshly isolated mouse trachea. This is accomplished by first preparing a shallow imaging chamber. The next steps of the procedure are to harvest and mount the tracheal tissue for imaging. It is then shown how to visualize and record cilia motility and cilia-generated flow using an inverted microscope with high-speed videography through a 100x DIC objective. The final step is to analyze the video by quantifying cilia beat frequency and cilia-generated flow using ImageJ software. Ultimately, the results can show changes in respiratory cilia motility and cilia-generated flow through high-speed video microscopy.
The main advantage of this technique over existing methods is that it allows reproducible imaging of the rodent tracheal epithelium for consistent quantitative assessments of ciliary motion. This can be accomplished within 20 to 30 minutes with minimal training. This method can help address key questions relating to the cell and molecular mechanisms regulating mucociliary clearance in the airway, especially when it is used in combination with different genetically engineered and mutant mouse models.
Begin by building the chamber used to hold the trachea samples. For a base or a chamber floor, use a glass-bottomed 35mm culture dish. For the top, use 0.3mm thick silicone sheeting. Lay the sheeting over an 18mm round glass cover slip and trim the edge. Next, with a scalpel,